Bismillah ar-Rahman ar-Rahim. This is a description of how to answer question 1 in Leave homework. He asked you to draw plan drawing for the lamina in the slide. The plan drawing should contain no cells, just layers. Here we have the upper epidermis, the cuticle, the palisade, the spongy according to the relative thickness of them, and we have here the lower epidermis thinner with the narrow stomata, the veins between the two layers divided into zones, and then he asks for the magnification of your drawing. Magnification equals image over actual. The image length is the length of this line drawn along the drawing, and you should draw this line and measure it. That measurement will be checked by the examiner. And then divided by the actual, which is the actual thickness of the lamina of the leaf, measured by the ruler under the microscope, and which will be equal 0.5 in this exam millimeter. So divide 85 millimeter over 0.5 millimeter to find the magnification. It should be a whole number. Question 2b, to draw the guard cells and the lower epidermal cells, two cells near these guard cells. Here the guard cells, you should take the shape and you should represent the thickness of the wall, thinner in this area, thicker in this area, and the lower epidermal cells surrounding with the cell wall two lines. Then he asks for the drawing of the upper epidermal cells should be wider and thicker than the lower epidermal cells with the cell wall. This is the answer for the question 2 in the homework. Here it is the exam of June 2008, variant 31. He asked to draw the fold of the maram grass. This is the outer epidermis, and then we have the shape of the fold. This is the photosynthetic tissue, the vascular body divided into two zones, and this is the inner epidermis. Don't forget to put some of the trichomes. And the hinge cells thin-walled, one or two closed lines, and they have the shape. The second part of the question asked you to draw the epidermal cells, outer epidermal cells. Here the cells should take the shape of the cells exactly as in the slide. The ratio between the length and the width, and two lines, and the cuticle very thick. Then he asked you to measure the base of the one of the trichome by the EPG, or eyepiece graticule. And it was found to be 1 EPG. It should be a whole number. Take care. And then you will calibrate the EPG with the stage micrometer under the same power, which was the high power, and it was found that 3 EPG equal 1 stage, which equals to 100 micrometer or 0.1 millimeter. This means then 1 stage equals 3, so the 3 equal the 100 micrometer, so the 1 equals 100 divided by 3 equals 33.3 micrometer. So this is the length of the base of the trichome, as it equals 1 EPG. So this is how to use the EPG after its calibration with the stage micrometer to find out an actual length. Bismillah ar-Rahim. This is the drawing for the slide in the third question. It is a pointed part of a leaf, and you should draw the exact shape pointed, and this is the epidermis, and this is the photosynthetic tissue. Above and below we have vascular bundles with connecting tissue, and we have here at the tip a few condensed cells which represent the cholenchyma. What are the requirements to take the full mark? Number 1, of course, the quality of the drawing, and this quality depends on the lines. The lines should be clear, single lines, not overlapping, not having a defect, no shading, and of course no cells are drawn, and the size of the drawing should be big enough so that you will take the mark for the quality. So the quality includes 3 requirements: the lines, the size, and no shading. And of course no cells, which is a character for the plan. Number 2, to take the mark, the tapering end of the leaf should be present because you are representing the exact shape of the lamina in front of you. 1 or 2 vascular bundles should be also present in your drawing. And of course, you have to draw the epidermis, as I have said, as 2 lines all through the lamina. Then he asked for annotations for visible features. Number 1, you can take the epidermis here. What are the features? They are one cell thick or thin wall cells. Another annotation for photosynthetic tissue, it contains here chloroplast. This is the answer for the second part of the question. He asks you to draw 2 epidermal cells and the cells underneath. Here the epidermal cells have a wall, and this is not a nucleus, it's an oil droplet, and these are palisade cells underneath. The wall is thin in all the cells. Here we don't have nuclei, here we have nuclei, we have also the chloroplast. Take care that if you label this black droplet as a nucleus, it's wrong, it's an oil droplet. And this is a nucleus, this is the air space, these are the chloroplast, this is the wall, and this is the epidermal cell. This is the answer for the question 4 in the homework. He asked you to draw a plan drawing for the midrib. Here we have concavity in the upper, convexity in the lower, a group of collenchyma cells and another group of cells. This is the vascular bundle, xylem above, and these are evident air spaces. Then detailed drawing for the 2 epidermal cells and 2 cells in the lower down 2 layers. Epidermal cells as usual, but take care that this middle layer, the cells are longest cells in the 3 rows. This one is not yet complete because of the end of the answer for the question 5 in the homework. He asked you to draw the plan drawing for the midrib of Nerium oleander and the detailed drawing for xylem vessels, now we will see how to draw this plan drawing, but take care that the wavy appearance, especially in the lower surface, is very important. And here we have some concavity at the upper surface. This is the epidermis and this is the cuticle. And we have here the area for photosynthetic tissue present, or the palisade tissue present in the laminae. And this is the vascular bundle. If you see the vascular bundle, you will see the xylem in the middle and double phloem layers here, which is very specific for the Nerium oleander. Then we have here the parenchyma tissue present at the base of the midrib. In case of the xylem vessels, as you see, they should have 5 to 6 edges with sharp corners, and the lumen here is very wide. In this line, you can make it more or less circular, not necessary to be with sharp corners as the outer surface. And this is, of course, the lumen. Now we will see how we will draw it. Here we have the midrib. Don't forget the wavy appearance lower down, the vascular bundle, the palisade on both sides. Here we have the parenchyma in the lower part of the bundle of the midrib. Then we have the 5 to 6 edges, very sharp corners, and here we have the lumen. This is the answer for the question 6 in the homework. He asks you to draw a plan drawing for the midrib. Here we have the upper epidermis, lower epidermis, the vascular bundle divided into 2 zones, and we have an area enclosed for the collenchyma, another area for the parenchyma lower down. We have also the appearance of palisade and the spongy separated by this line. So to take the full mark, you have to draw 2 lines for the upper epidermis and the lower epidermis, taking care that the upper is thicker than the lower. After that, you will take, this is 2 marks. We'll take a mark for the drawing of the vascular bundle in its correct position, divided into 2 or 3 zones. This is very important. Number 4, the enclosed areas which is present above or below or both of them. This is for collenchyma, this is for parenchyma mainly. After that, he asks you to make a label line for the xylem. So the xylem, it is the tissue which is present in the upper part of the vascular bundle. You should draw it, the line with the ruler. And then this line is very important to be drawn by the ruler, and then you write the label which is the xylem. To take also the full mark, you have to take care of the quality of your drawing. Quality includes the single clear lines, not overlapping, not shading, where a proper size of the drawing. This is the description of the second part of the question. He asked you to draw 3 xylem vessels in one chain and another 3 xylem vessels in another chain. They are similar to each other more or less, and they will appear in the following form. The first chain of 3 xylem vessels is this one, and you will draw also another chain in the following part of the question, but they are more or less similar. How you will take the marks? If you draw 3 cells for this chain, another 3 cells for the other chain, so the total of 6 cells, this will take 1 mark. The size of the cell at least not less than 2 centimeter or 20 millimeter across the largest cell, and each cell should touch one at least other cell, and at the area of touching you will have a middle lamella. Of course, you have very thick wall, and we have 5 to 6 edges with sharp corners, which is a typical feature for the xylem vessels. After that, he asked you to make a label line for the lumen. So you will make the label line by the ruler, not by hand, and then you will write the label lumen. Here is the answer for the question 7 in the homework. He asked you to draw a plan drawing for one lamina of rosemary leaf, which is one of the xerophytes. He is shading the area you want to draw, so stick to this area perfectly. The part to be drawn is curved like this one, and it has an outer epidermis, inner epidermis, trichomes, few trichomes can be seen. Also, we have this photosynthetic tissue here in this area. Photosynthetic tissue appears to be granular due to the presence of the chloroplast. And after that, he asks you to make 2 annotations to prove that it has the power to conserve water. Now I have 3 annotations ready. It has a thick cuticle to reduce water loss by transpiration as it is waterproof. Here it is 1 annotation. He asked for 1 annotation only, but I will tell you 3. 1 annotation, this one. Another annotation, it is folded to reduce surface area, to reduce water loss. Another annotation, the presence of trichomes to trap water vapor to decrease the water potential gradient between the inside and the outside of the leaf, so that reducing transpiration. Very important to mention 1 annotation. Stick to the number he asked to do in the question. And now you will see how I will draw it step by step. I should look carefully for the shape, for any curve in the shape of the lamina. This is the first thing. After that, the epidermis, outer and inner, then the photosynthetic tissue. And then I will make a label line for the annotation that I will mention. I will choose only 1 annotation from the 3 I have mentioned before. Either the trichome, or folded, or the thick cuticle. Each annotation should be described fully. This is the second part in the same question. He asked you about the detailed drawing for 4 cells under the vascular bundles, and each cell should touch at least 2 other cells. Now we will see how we will draw these cells. These cells, as you see, they are parenchyma cells. The parenchyma cells are characterized by being having 5 to 6 sides with rounded corners, not sharp corners like the xylem. They are rounded corners, and they should have air spaces in between and thin wall. So these are the criteria for them. And of course you will follow the instruction that each one should touch at least 2. So this is the picture of the detailed drawing for these parenchyma cells present under the vascular bundle. Take care to take the full mark. You should make a proper size for the cells, big enough. At least the largest cell should not be less than 40 millimeter in its largest dimension. Also, you can see here we have the air spaces, 5 to 6 edges, rounded corners, and the wall is thin wall, 2 lines, and at the neighboring condition we have a middle lamella. So this is the picture for the drawing, and he asked you to make a label line with the ruler for the cytoplasm. Try to put the label line more peripheral, not in the center, as in the center these cells have a vacuole, but you don't see the tonoplast at all. So try to make the labeling at the outer part of the interior of the cell. And by this you can take the full mark for the question. This is the answer for the question number 8 in the homework. He asked you to draw a plan drawing for the lamina of the leaf, and this leaf contains at least 2 vascular bundles and 2 canals. Here we have the epidermis and the photosynthetic tissue. It has a pointed tip. The vascular bundle should at least to be divided into 2 zones. It's a rule. And here we have the canals, which are open canals. So in order to take the full mark, you have at least 2 vascular bundles, and the vascular bundle is divided, and also you should have the region for the vascular bundle. The location of the vascular bundle should be proper and with the proper size in relation to the lamina. These should be taken in consideration. Also, you should draw the epidermis as 2 lines, and the area for the photosynthetic tissue as you see. Don't forget the quality. Here also take care that he asked you to draw a label line with a ruler for the xylem. It is present in the upper part of the vascular bundle. Don't forget that the quality has 1 mark. The quality includes 3 conditions. Number 1, the line should be clear, single line, no overlap, no defects. Number 2, the drawing has no shading. Number 3, the size of the drawing should be big enough and clear enough, filling almost all of the space allowed for the drawing. Of the question, he asked you to draw 4 cells forming a part of the resin canal. The cells, you will draw them according to their shape, their size, relative size, the ratio between the length and the width. They have thin wall, and they should have a curve as they are forming a part of a canal. First, mark for the quality. Number 2, 4 cells each touching one other cell. No more than 4 cells, no less. Number 3, the cell wall 2 close lines at the cell wall is very thin in the cells. And number 4 will be given if you have drawn the cell to form such a curve as they form a boundary of a rounded canal. So by this you can take the full mark for this drawing, which is a detailed drawing. Finally, he asked you to make a label line for the cell wall. Draw the label line. Here is the answer for the question 9. He asked you to draw detailed drawing for the 2 epidermal cells and 1 palisade cell underneath. Take care of the shape of the cells. The epidermal and the palisade both have thin wall. The epidermal cell has no contents evident, but the palisade has the chloroplast. If you see a nucleus, you can put it, but the chloroplast can be easily seen. This is the cell wall label with the ruler, and the cell wall is written beside this label line. The quality is von 1 mark, and the minimum size for the epidermal cells or to epidermals to the palisade cell will be at least 40 millimeter, so very important. And only you will draw 3, no more, no less. This is a mark also. And also you should draw the contents of any of the cells. Actually, the contents appear in the palisade only. No contents can be seen in the epidermal cells. And the label line with the ruler and write the cell wall. In this part of question 9 in the homework, you are asked to measure the diameter of the field of view using the clear plastic ruler. You will put the plastic ruler under the objective lens and measure the this diameter of the field. The diameter of the field was found to be 1 millimeter. It's found to be 1 millimeter. Okay, so this is 1 millimeter. Then after that, he asked you to measure the length of the diameter of view in the picture, which of course is magnified, and measure the length of ZY, which is occupied by the lamina of the leaf, and find the ratio between ZY and ZX. You will measure this and it will be 4 centimeter, so it will be 4 centimeter for ZY and 6 centimeter for XZ. So the ratio between this ZY to XZ will be 4 over 6. The fraction here will be 4 over 6. It's a ratio. Then he asked you after that you will use this fraction to find out the depth of the midrib shown by this line, the actual depth, the actual length of ZY. Now you can make it by multiplying the 1000 micrometer, which is the 1 millimeter, by this ratio. When it is multiplied by 4 over 6, you will find out the exact length of this part YZ. Then after that, he asked you that you will give your answer in micrometer, so it will be a whole number. Multiply this 1000 by 4 over 6, you will have a number with a fraction, with the decimal point. You will approximate it to have a whole number. You should have a whole number at the last. Then he asked you to find another way to measure this actual length. This will be by the eyepiece graticule, which will measure this length in eyepiece graticule and then calibrate the eyepiece graticule with the stage micrometer and find the value of 1 EPG. Multiply this value by the measurement you have measured by the EPG to find out the actual length of YZ.